Good morning, everybody. This is the first of three videos that I'm going to record for you guys on photosynthesis, which essentially makes life or makes life possible by using light and air, right? So I figured this was a good time to do it. It is a gloomy Sunday morning. It's about to get gloomier as the Bears play the Detroit Lions in a little bit that I'll be sitting down to watch. But before that happens, I thought I'd bring a little bit of information about the importance of light and how light is captured in photosynthesis, along with a couple other things about photosynthesis, so that we can really get into the nitty gritty details in the next two videos. So, um, photosynthesis and cellular respiration are connected and they kind of cycle nutrients through our biosphere, right? So heterotrophs make energy in organic molecules from ingesting them, right? So they are going to take glucose and they're going to oxidize it. They're going to require oxygen to do that, and they're going to generate carbon dioxide, water, and energy. And then they'll use that energy in the form of ATP to build their own organic molecules from the food that they eat, right? Um, so autotrophs, which are those organisms that can make their own food, make energy and organic molecules from light energy. Okay, so they're going to take in carbon dioxide, they're going to take in water, and they're going to use light, and then they're going to use all those things to generate uh, their own organic materials. And in the case of the photosynthesis examples that we're going to be looking at here, we're going to focus on the production of glucose. But if you're an autotroph, you have to be able to make all of your own organic molecules, not just glucose. They make all of their own organic molecules, and we'll talk a little bit about how they do that. And then they generate oxygen as a waste product. Okay, which for the plants is a waste product, but for us, it's not. Now, notice I said that autotrophs make energy, so you don't see any over here. They actually generate ATP in one part of photosynthesis called the light reactions, and then they will use that ATP in the second stage of photosynthesis called the Calvin cycle. All right, so photosynthesis converts light energy into chemical energy that is stored in sugar and other organic molecules. And so you'll see here in this diagram, they're going to use the light energy is going to, the light reactions are going to use light energy and water. And then the light reactions is where oxygen gas actually gets generated as a waste product. The light reactions are going to generate ATP and an electron carrier molecule called NADPH, which is a little bit different than the NADH of cellular respiration, but it's just a small little difference of an addition of a phosphate group that causes it to be called NADPH. You can remember the P for photosynthesis or P for plants, um, but then and then the ATP and NADPH, those electron carriers generated in the light reactions, are used in the second stage of photosynthesis called the Calvin cycle, along with carbon dioxide to generate sugars and other organic molecules. The ATP that's used is recycled back as ADP plus a phosphate group, and then ADPH drops off its electrons and is recycles NADP plus, which is able then to pick up electrons. So autotrophs make organic molecules from carbon dioxide and inorganic materials in the environment, like they're going to require some magnesium and some other things. And they are considered to be producers, right? So they are producing organic molecules that can be consumed by heterotrophs, right? That live on the organic molecules made by other organisms. So consumers and decomposers both are heterotrophs. Okay, so in order to be a plant, you have to be able to collect light energy and transform it into chemical energy, right? In the form of ATP. You have to be able to store that light energy in a form that can be moved around the plant or stored, right? So once you convert it, once you collect it, you've got to convert it into a form that can be stored. And you're actually going to convert that and store that light energy up in glucose that can be moved around the plant. And you have to get your building block atoms from the environment, right? Somehow you've got to get carbon dioxide, you've got to get water, and you don't do it by ingesting it. Okay, so you need carbon, hydrogen, oxygen, nitrogen, all those nutrients, all those atoms you got to get from the environment somehow. Okay, and so that is because as a plant, you have to produce all your own organic molecules. You don't consume any of them. So plants are going to get their carbon and oxygen from carbon dioxide. They're going to get their hydrogen from water, and they're going to get all these other minerals dissolved in the water that they absorb. Okay. So plants produce all of their own organic molecules, right? Carbs, proteins, lipids, nucleic acids, they make their own. So where do they gather all of these materials from, right? Sunlight is gathered by the leaves, right? The leaves of a plant are basically like little solar panels or solar collectors for the plant. Carbon dioxide is also uh, gathered by the leaves through the stomata, which we looked at during our cell unit. We saw the stomata in the leaf cross section. Those are the openings on the underside of the leaf that allow for gas exchange. They allow carbon dioxide in and oxygen out. Water is gathered by the roots, and so are most of the other nutrients gathered up by the roots as they are dissolved in the water that the roots take in. Okay, so chloroplasts are where photosynthesis takes place, right? So we're going to kind of zoom in on the chloroplast here. I do want you to remember that plants are not the only photosynthetic organisms. We have unicellular protists. We have purple sulfur bacteria that are photosynthetic. Cyanobacteria are believed to have been the first organisms to have actually carried out photosynthesis on the planet. So photosynthesis actually evolved in prokaryotic organisms. The proteins necessary for photosynthesis would have been embedded in the plasma membrane of those cyanobacteria. And it's believed that they generated the oxygen that is a major part of our atmosphere now. And then there are multicellular algae also, like kelp, for example, that carry out photosynthesis. Okay. So as we look at a plant and we try and figure out where the chloroplasts are, we're going to kind of review the cross section of our leaf right here. We have the upper and lower epidermis that just kind of serve as protection. The top layer of cells here is called the palisade mesophyll, all tightly packed together. And the palisade mesophyll is loaded. These cells here are loaded with chloroplasts. We have the spongy layer or spongy mesophyll here. All these cells with gaps between them are also loaded with chloroplasts, but there's air spaces between them to allow carbon dioxide gas to diffuse throughout the plant leaf. There's also going to be a vascular bundle or a vein within the leaf that contains xylem cells that have these thick walls that are bringing water up to the leaf, and phloem cells here with thinner walls that are carrying the sugars and other organic molecules that are made in photosynthesis away from the leaf to other parts of the plant. And then the lower layer, the lower epidermis here, has these openings called stomata that have guard cells on either side of them that allow CO2 in and water and oxygen out. Okay, so you can see all those other things located here. The top layer often does have a waxy layer called a cuticle on top of it that does help to prevent water loss from the surface of the leaf. Okay, and then transpiration and gas exchange takes place through the stomata. And one other thing I want to point out, the cells in the top layer of the leaf are all tightly packed together, loaded with chloroplasts. It makes sense that you would have the high concentration of photosynthetic cells on the upper surface of the leaf, because that's where light is coming down and hitting the leaf, is that upper surface. So this is an example of that one theme we've talked about many times, that form fits function, right? And then having these air spaces down here where gas is coming in through the lower part of the leaf also is another example of form fits function, because they allow for gases to diffuse throughout the leaf then after they come in. All right, so if we take a look at chloroplasts, we're going to see that the mesophyll cells are loaded with chloroplasts, and we're going to talk a little bit soon about the structure of the chloroplast. One thing you should note is that the leaves with these chloroplasts absorb sunlight, okay, and the leaf also and then they also will take in CO2 that the leaf has absorbed. Now, some of the wavelengths of light are absorbed by these chloroplasts, and they're specifically absorbed by pigments within the chloroplast, such as chlorophyll, which is the main photosynthetic pigment, but not the only one. And then some of the wavelengths of light are either reflected back to our eyes or transmitted through the chloroplast, right? And that's why plant leaves look green is because the green wavelength of light is what is reflected by the pigments in the leaf. All other wavelengths of light are going to be absorbed by the pigments in the leaf. Okay. All right, so let's take a look at a chloroplast, right? It is a double membrane structure, which again, that double membrane supports the endosymbiotic theory because it's believed that the chloroplast was a free living photosynthetic prokaryote that was engulfed by a larger prokaryote. So that's where this second membrane comes from, is from that phagocytosis. Uh, Chloroplasts, so they have that outer and that inner membrane. They also, just like mitochondria, have their own DNA and their own ribosomes, and their DNA is circular. The fluid-filled interior of the chloroplast is called the stroma, which you see there. There's all these little discs stacked on top of one another within the chloroplast. Each of these discs is called a thylakoid or a thylakoid sac. And the thylakoids are stacked, and the stacks of thylakoids are called grana. An individual stack is called a granum. Okay. The thylakoid membrane itself contains chlorophyll molecules and other pigment molecules. It contains an electron transport chain and contains the enzyme ATP synthase for making ATP. So let me move this here. So there's our chloroplast with its double, and then it's got a double membrane. And then inside, we're going to have all these thylakoids, which are going to have a high concentration of protons within the thylakoid sac. And just like in cellular respiration, that proton gradient is going to be used to drive the production of ATP by activating ATP synthase. Okay, so it's going to be very similar. All right, so photosynthesis, two stages: the light reactions, which are also called the light dependent reactions or the energy conversion reactions, because they convert solar energy into chemical energy of ATP and NADPH. Okay, and the NADPH, just like NADH, is an electron carrier that has a lot of stored energy that could be used later. The second stage of photosynthesis is also called the Calvin cycle or the light independent reactions. It is not, however, it is not called the dark reactions because it cannot take place without the products of the light reactions. So when there is no light, the light reactions aren't taking place, so the Calvin cycle cannot take place either. Okay, so in the Calvin cycle, sugar is built. So the chemical energy generated in the light reactions, the ATP and the NADPH, is used to reduce carbon dioxide and make glucose. Okay, the Calvin cycle, by the way, takes place in the stroma, whereas the light reactions take place within the thylakoids. Okay. So the light reactions are going to have an electron transport chain, just like in cellular respiration. The proteins are going to be embedded within an organelle membrane. However, they're embedded within the thylakoid membrane. Okay. It involves electron acceptors, right? NADP plus is the electron acceptor that is reduced to NADPH, which carries those electrons around. It involves the buildup of a proton gradient across a membrane. And then it involves an ATP synthase enzyme. Okay, so just like we talked about before here. Okay, so we'll talk in detail about that. So here's the electron transport chain of cellular respiration. Remember, we transfer the chemical energy from food molecules into the chemical energy of ATP through oxidative phosphorylation using the energy carrier NADH, right? Okay. Here in photosynthesis, the chloroplasts are going to transform light energy into the chemical energy of ATP using NADPH, and oxygen is going to be generated in this case where water was generated in the electron transport chain of cellular respiration. And we're going to get into the details of all this later. Don't worry about if you don't understand exactly everything that's going on here right now. This right here was just to point out we're transform light energy into ATP, and we're going to generate an electron carrier, NADPH. So photosynthesis uses sunlight. Respiration uses the oxidation of glucose or the breakdown of glucose. But in both cases, the energy from either sunlight or the oxidation of glucose is used to move electrons. So we can run a pump that pumps protons into some sort of space where a proton gradient is built up. And so The protons are then going to flow down their concentration gradient through ATP synthase, which bonds phosphate groups to ADP to generate ATP. Okay. Now, remember this, the generation of ATP in cellular respiration in this manner was called oxidative phosphorylation because of the energy to create ATP, the energy to phosphorylate ADP to create ATP came from the oxidation of glucose. In photosynthesis, the generation of ATP uses the energy from sunlight to pump electrons so that proton gradient can be built up so we can phosphorylate ADP. So this type of phosphorylation that occurs in the light reactions of photosynthesis is called photophosphorylation. So the energy that drives the phosphorylation of ADP comes from sunlight, hence photo, right? Photo meaning light, so photophosphorylation. Now, photosynthesis involves several different pigments. The main pigment of photosynthesis, the primary pigment, is chlorophyll A. There are a few different types of chlorophyll. They differ very slightly. You can see in chlorophyll A right here is a methyl group attached to this carbon. It is an aldehyde that's attached to this carbon in another version of chlorophyll called chlorophyll B. All right, but the chlorophyll molecule here that captures light energy actually requires a magnesium ion to be centered at the middle of the molecule here, at the light absorbing head of the molecule, that is going to be embedded in a protein, right? These pigment molecules all get embedded within proteins that are found within the thylakoid membrane. Okay, and then we have this long hydrocarbon tail that extends down into the hydrophobic portion of the protein, which kind of makes sense here. So chlorophylls and other pigments are embedded in the thylakoid membrane, and they are arranged into what are called photosystems, which are these collections of proteins and pigment molecules all clustered together, creating these larger complexes called photosystems. Okay. And there are actually two different photosystems in most eukaryotes that we'll talk about later. Okay. Now, when chlorophyll is struck by light, okay, the light energy actually excites electrons within that chlorophyll molecule. And when electrons get excited, you might remember from chemistry, they jump to a higher energy level, but that is a highly unstable configuration for those electrons, and they usually release that energy very quickly and crash back down to their original ground state. And when they come back down, they release heat energy and the light energy, and the wavelength of light is equivalent to the amount of energy that they absorbed. So if we actually take chlorophyll and remove it and make a chlorophyll solution by removing it from plant leaves, which I've done in the past and could do if we'd been in class here, we create a solution of chlorophyll, and if we shine a black light on it, it actually fluoresces here as the electrons get excited and then crash back down to their ground state, releasing wavelengths of light, releasing energy that we see as those wavelengths of light. Now, plants, though, if you shine a black light on them, they don't fluoresce like this, because once those electrons get excited within a functional chloroplast, those excited electrons get captured by other molecules in that high energy state. Okay, so quick reminder as we look at the wavelengths of light here, what plants actually capture among the electromagnetic spectrum is the visible light spectrum. So that we're talking about the shorter wavelengths to the longer wavelengths of light here. We're talking about Roy G. Biv. Okay, Mr. Biv, I'm sure you remember learning about Roy G. Biv. The red wavelengths of light have the longer wavelengths, which means they actually have a little bit lower energy. And the violet wavelengths of light have the shortest wavelengths that are absorbed by plant leaves and have the highest amount of energy. Okay, I love this little comic here. You can pause it if you want to read it, but I will keep going. But this is one of my favorites here. So when we talk about the light absorption spectrum, right, which wavelengths of light are absorbed and how much are absorbed? Okay, photosynthesis gets its energy by absorbing different wavelengths of light, and there are several different pigment molecules that do this. Chlorophyll A is the main photosynthetic pigment that absorbs best in the red wavelengths and blue wavelengths of light, and the least in the green. There are accessory pigments that absorb slightly different wavelengths of light to help plants capture almost the entire spectrum, visible light spectrum. So very little energy is not trapped by these chlorophyll molecules and other pigment molecules as light hits these plant leaves. Okay. There are pigment molecules called carotenoids. There are pigment molecules called xanthophylls that are helping to absorb these different wavelengths of light. They also help to play a little bit of a photoprotective role, because all of this energy all this light energy striking the plant could certainly damage the cells of the plant. And so by absorbing some of that light, it also helps to protect the plant. Okay, so your other pigments besides chlorophyll A, which is your primary photosynthetic pigment, are referred to as accessory pigments. And chlorophyll B, carotenoids, xanthophylls, those are all examples of pigments that absorb slightly different wavelengths of light. Okay. There are other pigments called anthocyanins that are actually kind of a reddish blue. So there's a couple ways that we have determined how which wavelengths of light plants absorb. So we can take white light and shine it through a prism so that it refracts and generates all of the entire wavelength of all. We can see the entire visible light spectrum, and then put different filters in front of the uh, in front of those different wavelengths of light, so that we allow only a selected wavelength to pass through, for example, green light. And then we can measure how much of that light is absorbed or how much is transmitted and reflected by using different tools like spectrophotometers that measure how much of a particular wavelength of light is absorbed by a solution. And what we can then create is what's called an absorption spectrum for each different pigment molecule, right? We can isolate the different pigment molecules, put them into these test tubes, and allow the different wavelengths of light to strike them, and then measure how much of that light is being absorbed, how much is being transmitted. And we create these absorption spectra where we see which wavelengths of light are absorbed the best by these different pigment molecules. Where we see these peaks would tell you which wavelengths are absorbed the best. Now, then what we can also generate is what we call an action spectrum, where this is different. We're not measuring the absorption of a particular pigment. We're measuring how much photosynthesis is taking place, right? How much action is actually taking place. And we measure that by either measuring how much oxygen is being released or how much carbon dioxide is being removed, right? And we see that the absorption or the action spectrum that we see here kind of mirrors the absorption spectra of these different pigment molecules, in that there's a lot more oxygen generated at the violet and blue wavelengths of light. There's very little oxygen generated at the green and yellow wavelengths of light, and then a lot more again at the red and the orange. Okay, and there was one experiment carried out by a Dutch, I think he was a Dutch scientist in the 1800s. His name was Johann Engelmann. And he took a filamentous algae, and you should recognize this. This is Spirogyra is the name of this one. And he took some white light, put a prism in front of it, and then shined that light onto this filament of algae, so that it had the algae at one end had like the red and oranges striking it, and here in the middle, greens and yellows, and then on the other end of that filament, there were the blues and violets, right? And then what he did was he took a culture of aerobic bacteria, put them on the same slide with this algae that had the visible light spectrum shining on it, and looked to see where the aerobic bacteria that require oxygen in order to survive actually congregated. And they clustered at the red and orange end, and then they clustered at the blue and violet end. Very few were found in where the green and yellow wavelengths of light were located, because here the Spirogyra with the chloroplasts inside were carrying out very little photosynthesis, so very little oxygen was being generated here in this area with the green and yellow wavelengths striking the algae, whereas a lot of oxygen was being generated in the red and orange areas and in the blue and violet areas. And the pattern that these bacteria congregated kind of matches the action spectrum for photosynthesis. This is a really cool experiment. It is very elegant experiment, and very imaginative and creative, for somebody who didn't have all the modern tools like we have to measure oxygen production and to measure absorbance. So anybody who says that scientists aren't creative, they're not accurate. Okay, so that's where I'm going to stop this particular lecture. We will get into the photosystems of photosynthesis and how photosynthesis, the light reactions of photosynthesis work in the next video.